All right, welcome everyone. I am your moderator, Katherine Brendish. I'm a clinical professor and the program director for the Genetic Counseling Master's Degree Program at Arizona State University. Prior to joining ASU in 2019, I worked in clinical care as a genetic counselor at the Mayo Clinic, where I specialized in hereditary cancer, neurological, and cardiac conditions. I'd like to thank you all for joining our Health Talks webinar today, brought to you by the College of Health Solutions at ASU. Our college works to address the challenges facing people and communities to stay healthy, improve their health, and manage chronic disease. These Health Talks are one way we serve the community with timely and relevant educational information that also provides continuing education credit at no cost. Before we begin, I have a few quick housekeeping items. First, this session is being recorded and will be posted on our website, asuhealthtalks.com. After the panelist presentations, I will moderate a Q&A session. We'll have about 20 minutes for questions and answers, and please submit your questions using Zoom's Q&A feature and not the chat box. Regardless of whether or not you've registered for CEUs, please register your attendance using the self-check-in link posted in the chat, and Carl just posted that for us, so we have an accurate record of who attended today's webinar. And finally, you will receive a brief survey asking for your input about today's webinar. If you are requesting continuing education credits, this must be completed in order to receive credit. If you did not apply for CEUs, we would still appreciate your feedback to improve our series. All right. I'm really excited now to move on and introduce today's speakers. First up is Keri Ramsey. Keri is a genetic counselor, a nurse, and clinical co-director of the Center for Rare Childhood Disorders at TGen, a nonprofit research institute in Phoenix. With a background in genetic counseling, nursing, and neuroscience research, she has worked in both critical care and clinical genetics, focusing on rare childhood disorders and pharmaceutical trials for conditions like Rett syndrome. Keri will be followed by Dr. Narayanan. Dr. Narayanan is a child neurologist and medical director of the Center for Rare Childhood Disease at TGen. We are so fortunate to have our health experts with us today as we explore rare genetic disorders, targeted support from diagnosis to therapy. Rare genetic disorders, once difficult to diagnose, have seen significant advancements through genomics, enabling precise diagnoses and the development of targeted therapies, as demonstrated by the successes in treating conditions like spinal muscular atrophy and Duchenne muscular dystrophy. Today's presenters will provide an overview of the prevalence of rare genetic disorders and the current diagnostic approaches, while also examining the broader implications for treatment and patient care. So please welcome our first presenter, Keri, who will talk about rare genetic disorders, targeted support from diagnosis to therapy. Thank you, Keri.
Thank you, Katherine, for inviting us to speak today. I'm going to go ahead and share my slides. Okay. As Katherine mentioned, today I'm going to speak about rare genetic disorders, targeted support from diagnosis to therapy. These are my disclosures. So first, I want to give a brief introduction to the Center for Rare Childhood Disorders, or sometimes we call it C4RCD, and its contributions to rare disease research. Then I'm going to explore the meaning of rare diseases. We're going to talk a little bit about next-generation sequencing and its impact on diagnosis. And then we're going to learn about current and emerging treatment modalities for rare diseases. So TGen is a nonprofit research institute in Phoenix, Arizona. We're also a part of City of Hope. The Center for Rare Childhood Disorders was founded in 2012 with a mission of providing diagnosis for individuals with rare disease. Dr. Vinodh Narayanan is our medical director, and as Katherine mentioned, he'll be speaking after me. Dr. Matt Hunnelman is our scientific director. We have two clinical research coordinators, Meredith and Newell, two research associates, Anna and Jennifer, and then our bioinformatician, Marcus. And I just wanted to say thank you up front to all the people that make up the center and all the hard work that they do every day. So the C4RCD has provided access to next generation sequencing for over 750 rare disease families. And this is at no cost to the families. At no time have they paid a dime for our services. We have about 70% of our patients from Arizona, although we can enroll from anywhere in the United States. And we even have a cohort of patients from Hermosillo, Mexico that travel up to see us here in the clinic for enrollment. We have around about a 40% diagnostic rate. We've obtained about around over 200 patient cell lines. And this is typically fibroblasts that are taken from skin biopsies. And we've shared these with over 30 institutions around the world. And what's really great about these fibroblast cell lines is that we can convert them into induced pluripotent stem cells, which are cells that we can treat with chemicals to turn into different types of neurons, cardiac or muscle cells, depending on the type of functional work that we want to do in the lab. We've also participated in multiple clinical trials, including in studies for tuberous sclerosis and Rett syndrome. And Dr. Narayanan will go into a little bit more detail about Rett syndrome. So what is a rare disease? In the United States, it's defined as affecting less than 200,000 individuals. In Europe, they say no more than one person in 2,000. There's also a term ultra rare that has widely been used, and this is an incidence rate of less than 1 in 50,000. There's also hyper rare, which is used to describe diseases that affect 1 in 100 million people. So that would be about 80 or fewer people worldwide. And interestingly enough, we do have a couple of patients at our center who we would consider having a hyper rare disorder. There are estimated to be over 10,000 rare diseases, and this includes cancers and disorders that currently do not have a gene associated with them. And 1 in 10 individuals are affected by rare disease. It's thought that 80% of rare disorders are caused by gene mutations or variants. And on average, it takes over 6 years for a rare patient to receive an accurate diagnosis. 1 in 2 patients is a child, and about 45% of rare diseases have some neurological component. In terms of cancers, 20 to 25% are considered rare, and 95% of rare diseases lack an FDA-approved treatment. This is a graph that I pulled from the online Mendelian Inheritance in Man website. This is a database for genes and genetic disorders. And what this graph shows is that since the completion of the Human Genome Project in 2003, we have been able to discover new gene disease associations at an alarming rate. And this continues to happen every year. And this is largely in part due to efforts such as Gene Matcher, which is a freely accessible website developed at Baylor Hopkins Center for Mendelian Genetics. And I always joke and say this is like a matchmaking site for geneticists. Essentially what happens is you go onto the website, and if you have a patient who has a variant in a gene that has not been previously associated with human disease, then you can put that information on their website. If there's another clinician or researcher who has a patient who also has a variant in that same gene, you'll receive an email that you've matched. And then essentially you take the communication offline and you share information about the phenotype or clinical symptoms of your patient. And for our center, this has led to multiple collaborations and multiple new gene disease associations. And from the graph down below, you can actually see the number of publications that cite Gene Matcher. The orange bar represents new disorders, gene disease associations, and this is only going up until 2021. This website's been around since 2013. They have over 16,000 submissions from over 114 countries. And so I just wanted to provide just a general description of genetics, because I know we have a lot of people on the call, the presentation that are from different backgrounds. And essentially in our bodies, we have trillions of cells, and our genetic material is packaged in chromosomes that are in the nucleus of those cells. On the bottom right-hand corner, we see an image of a chromosome that's being unraveled. And along that stretch, we see the genes and the DNA, which is represented by that double helix image. That DNA is made up of 4 nucleotides represented by the letters A, T, G, and C. And if we look at a gene, which is at the very top, this is our DNA, a gene is made up of both exons and introns. And the exons are the part of the DNA that codes for protein in the cell. And you see here through the process of transcription, the introns are actually spliced out to create messenger RNA. That messenger RNA is then translated into an amino acid chain, which is folded to form the proteins. And now I want to talk about 3 different types of next generation sequencing that are being applied to help diagnose rare disease patients. And the first that I want to talk about is probably the most common type, which is exome sequencing. And you can see in the top right-hand corner, with exome sequencing, we're primarily focusing on the exons and the region flanking those exons, which is into the intronic region. And this is only about 1 to 2% of our DNA. And what happens with exome sequencing is that the DNA is fragmented into 50 to 300 base pair read lengths. And then these read lengths are aligned to the human genome reference. This type of technology is often covered by insurance, especially for conditions such as developmental delay, intellectual disability, multiple congenital anomalies, hearing loss, autism spectrum disorder, or epilepsy. And of the 3 I'm going to discuss today, this is probably the least expensive. Next is short read genome sequencing. And you can see again in the top right corner that not only are we looking at the exons, but also the regulatory regions and the non-coding regions, such as the introns. This covers about 92% of the genome. And again, we're looking at these 50 to 300 base pair read lengths. This type of technology is covered by some commercial insurances and is more expensive than exome sequencing. And then lastly, I wanted to talk about long read genome sequencing. And this is almost 100% of the genome. This includes not only the exons and introns, but also tandem repeats. So examples of this would be short tandem repeats in Fragile X syndrome or Huntington's disease, interspersed repeats, DNA transposons, telomeres, so the ends of the chromosomes, pseudogenes. These are genes that have very similar sequence homology to other genes, complex structural rearrangements, and then methylation status. And the reason that we can do all of this is because the read lengths in long read are typically around 10,000 to 100,000 base pairs in length, sometimes even being reported up to 4.2 megabases. At this point, it's mostly research-based, and of the 3 technologies is the most expensive. So I wanted to highlight these 2 papers. They were published last year and were out of the result of analyzing the 100,000 Genome Project in the UK. And what both of these groups found is that variants in a gene called RNU4-2, which encodes a critical component of the spliceosome, is now one of the most common autosomal dominant causes of intellectual disability and developmental delay. And this is a gene that up until now was not being sequenced with exome sequencing because it lies within the non-coding region of the genome. And one of the studies even said that this gene was the second most prevalent etiology for neurodevelopmental delay after MECP2, which is the cause of Rett syndrome. So it just shows that there's just still so much of the genome that we don't even understand or haven't associated with human disease at this point. I also wanted to share with you briefly a case report of 2 brothers who were referred to our clinic with suspected Duchenne muscular dystrophy. The proband was an 8-year-old male patient with pain, frequent falls, and difficulty walking upstairs. He'd fractured his femur at 6 years of age after tripping on a curb, and after extensive physical therapy was still not walking. He had been referred to a neurologist who found that the child had a CK level of over 12,000, which is highly elevated. A muscle biopsy showed negative dystrophin staining, and he was suspected to have Duchenne muscular dystrophy. He was started on steroids, and multiple genetic tests were run, which were all negative. This included a neuromuscular gene panel, exome sequencing, chromosomal microarray, and RNA sequencing of his muscle biopsy tissue. This family was referred to our center for whole genome sequencing. And you can see on the right in the pedigree, we not only enrolled the proband, but also his half-brother, who at 15 months also had an elevated CK level, his mom with a slightly elevated CK level, and both of their fathers. There was no significant family history for Duchenne muscular dystrophy. And interestingly, what we found was that the mom and both boys actually had an inverted insertion of 306 kilobases from chromosome 10 within the intron of the Duchenne muscular dystrophy gene, intron 44. And here is an image showing that both boys had intact copies, 2 copies of chromosome 10. They had inherited a Y chromosome from their father, and then they had inherited this chromosome X that had this insertion of about 6 genes. Of these 6 genes, only one of the genes is actually associated with a Mendelian disorder, and it's an autosomal recessive disorder associated with an immune disease. We followed up this genome sequencing by performing our own internal RNA sequencing of muscle tissue and found that the insertion of this part of chromosome 10 actually caused a fusion transcript between the PFKFB3 gene, shown in green here at the bottom, to exon 45 of the dystrophin gene. So this was definitely the cause of muscular dystrophy in both of these boys. And what's significant about this case is that Duchenne muscular dystrophy has multiple FDA-approved gene therapy treatments, and the treatment is very dependent on the type of mutation or variant that the patient has. And because we were able to identify the cause of DMD in these patients, they now are eligible to participate in one of the gene therapies. I also wanted to talk about the Guardian study, and this is a collaboration between one of the major genetic testing companies and Dr. Wendy Chung's group out of Columbia University. And this study aims to screen 100,000 newborns with genome sequencing, so newborn screening with genome sequencing. To date, they've performed this on over 15,000 newborns, and they're looking specifically at 450 conditions. Some of the key findings thus far have shown that 3.7% of these infants had positive screenings. The majority of those would not have been picked up with traditional newborn screening today. And just to give you some context, most states screen for about 30 to 50 conditions. Some of these conditions that they identified were long QT syndrome and Wilson's disease. Around 72% of families they approached for the study consented to participate, and over 90% of families requested inclusion of screening for optional neurodevelopmental disorders. There's also a similar study in rabies called Baby NSG, which is also looking at newborn screening. I also wanted to talk briefly about rapid genome sequencing and its application in the neonatal or pediatric intensive care unit. With this type of sequencing, the results are typically returned within 5 to 8 days. And the idea is that a child or an infant will receive a genetic diagnosis and hopefully treatment before they leave the hospital. 10 states have adopted rapid genome sequencing, including Arizona as of last year. The paper that I've highlighted here was written by Kingsmore and colleagues, and they reviewed 44 studies of children in the ICU who had rapid genome sequencing. They found that 30% of these individuals had received a diagnosis, 26 had changes in their treatment, and the net healthcare costs were reduced by $14,000 per child. So why is having a genetic diagnosis important? The first is that it has the possibility of guiding treatment options. An example of this is in individuals with GLUT1 deficiency who have seizures and can benefit by being placed on the ketogenic diet. It can also help with increased cancer screening, such as detection of Wilms tumor and a hepatoblastoma and Beckwith-Wiedemann syndrome. It can help confirm a specific diagnosis and thus end the diagnostic journey for these families. It can also help them participate in clinical trials, which is a perfect example of the Duchenne muscular dystrophy family that I just talked about. It can prevent or limit unnecessary treatment or invasive procedures and their associated costs, such as muscle biopsies, MRIs, EMGs, and surgeries. It can help predict the course of a disorder through natural history studies, and in some cases can help families with an estimated lifespan for their child. It can help with family planning in calculating recurrence risk, pre-implantation genetic testing, CVS or amniocentesis, and testing of family members. It can also help families participate in social networking, such as Facebook groups or different foundation websites, and some of these groups even have annual family meetings. And lastly, the creation of patient registries. Some examples are CORDS or RareX, and it's estimated that there are around 845 registries worldwide. So now I want to switch and talk about 3 of the most common types of treatment for rare genetic diseases. This includes small molecules, enzyme replacement therapy, which is either using natural forms or recombinant proteins that show a high degree of homology with human enzymes, and then also gene therapies, such as RNA therapy, where a shorter sequence of RNA material is used. This includes antisense oligonucleotides, mRNA, si, and tRNAs, and this typically involves gene silencing. There's gene addition, where a functional copy of the gene is delivered via a vector, such as adeno-associated viruses, or retroviral or lentiviral vectors. There's also gene-modified cell therapies, where you first remove a patient's own cells from the body, such as hematopoietic stem cells. Then these cells are modified by either adding a working copy of a gene or by modifying the affected one. And then these cells are then returned back to the person. And lastly, gene editing, which corrects a piece of a patient's DNA by changing or deleting their own DNA, such as CRISPR-Cas. So for small molecules, I want to talk about mTOR inhibitors for tuberous sclerosis complex. TSC affects roughly 1 in 6,000 to 10,000 individuals. It's a multi-system disorder that affects the nervous system, the eyes, the heart, the lungs, liver and kidneys, skin, and nails. And it's caused by variants in the TSC1 and TSC2 gene that affect the mTOR pathway. Everolimus, which is an mTOR inhibitor, has been approved for the treatment of TSC-related complications. And this includes SEGAs, which are subependymal giant cell astrocytomas. These are benign tumors in the brain that can cause hydrocephalus due to the buildup of cerebral spinal fluid. Angiomyolipomas, which are found in the kidneys. And this medication has also been found to decrease seizures in patients. When we think about enzyme replacement therapy, we think about lysosomal storage disorders. And the first approved ERT was for Gaucher disease in 1991. This type of therapy is usually administered through IV infusion every week or every other week. It's important for it to be delivered before irreversible tissue damage occurs, and the enzymes cannot cross the blood-brain barrier. I've shown on the right here an article describing the first successful in utero treatment of Pompe disease. This little girl, unlike her previous siblings, did not have the development of cardiac hypertrophy. She had lower creatine kinase levels than age-matched patients and demonstrated normal muscle function. And there are currently still clinical trials going on for multiple lysosomal storage disorders in utero treatments. Probably one of the most well-known applications of gene therapy is for spinal muscular atrophy, which is a rare genetic disorder that affects the motor neurons in the spinal cord. The most severe type, type 1, if left untreated, can cause death in the first 2 years of life. 95% of SMA is caused by a homozygous loss of exon 7 in the SMN1 gene. In the picture below, on the left, we see a healthy individual. On the right, we see a patient with SMA who is not producing SMN from the SMN1 gene. However, there is a gene called SMN2, which is a paralog of SMN1. There is a C to T synonymous nucleotide change in exon 7 of SMN2 that affects splicing and results in very low levels of full-length SMN2 transcript. You can see in the bottom right-hand corner of the image that this particular individual is producing around 10% of the full-length SMN protein. And a person can have a variable number of SMN2 copies, typically ranging from 0 to 8. And this number can be roughly correlated to the severity of symptoms in patients with SMA, those having more copies of SMN2 having a less severe clinical presentation. And here are the 3 FDA approvals for SMA. The first was nusinersen, which is an antisense oligonucleotide designed to increase exon 7 inclusion in the SMN2 mRNA transcript. And what this does is results in the production of full-length SMN. This is an intrathecal injection given every 4 months, and it's eligible to all ages. Below that, we have risdiplam, which is a small molecule that modifies SMN2 pre-mRNA splicing and again promotes the inclusion of exon 7. However, this is an oral medication that's given daily, and it's eligible to all ages. Lastly, we have on the bottom Zolgensma, which is a gene replacement therapy that uses an adeno-associated virus carrying a self-complementary SMN1 gene, cDNA. And this is administered via single dose, IV dose, and is given to individuals less than 2 years of age. In this particular case, early diagnosis is important, and that the treatment is given before significant motor neuron loss. And so for that reason, SMA has been added to newborn screening in all 50 states. And lastly, I wanted to talk about gene editing with sickle cell disease. Casgevy was the first CRISPR-Cas9 therapy approved by the FDA in 2023. It works by modifying a patient's own hematopoietic stem cells, making them produce more fetal hemoglobin. This prevents sickle cells by reactivating a gene that normally shuts off after birth. And this is performed by taking a patient's own blood cells, and you can see this in the image on the right, editing them outside of the body, and then reinfusing them back into the patient where they begin to produce the modified red blood cells. And in this case, CRISPR-Cas9 functions by utilizing a short RNA segment known as a guide RNA that directs the Cas9 enzyme to a specific site on the DNA strand. Once there, the Cas9 cuts the DNA, which enables scientists to precisely edit the genetic code by either inserting, removing, or altering the DNA sequence at that specific location. In the case of Casgevy, it deletes the enhancer that controls the expression of the BCL11A gene, which suppresses fetal hemoglobin production. This particular therapy is available to individuals that are older than 12 years of age with vaso-occlusive crisis. This improves red blood cells and can eliminate painful crises and the need for blood transfusions. Casgevy and the other gene therapy that were approved for a CD are listed at $2.2 million and $3.1 million. The FDA's Center for Drug Evaluation and Research, or CIDR, approved 26 novel therapies for rare disease in 2024. In 2023, there are around 28 novel therapies. The diseases without previous treatment included Niemann-Pick type C and WHIM syndrome, but also included gene therapy for AADC deficiency, hemophilia type B, and metachromatic leukodystrophy. And that concludes my talk. I just wanted to remind everyone that next Friday, February 28th, is Rare Disease Day. And you can visit the website at the bottom to find out more information on how you can celebrate this special day. I also wanted to thank all of the patients that are part of our center and all the collaborators that have worked with us, and provide my contact information in case you wanted to follow up with me after the talk. And now I'd like to hand it over to Dr. Narayanan.
And I'll just interrupt real quick with a comment. Thank you, Keri. That was fantastic. That was such a wonderful overview and a very nice setup for our next speaker. Before I introduce our next speaker again, I just want to remind everyone, if you haven't already done so, please register your attendance using the self-check-in link posted in the chat so we can get an accurate record of who attended today's webinar. All right, as promised, now we'll hear from Dr. Narayanan, who's going to discuss Rett syndrome, a bird's eye view.
Okay, let me make sure I get through the technical hurdles. Just lost where my Zoom connection is.
You may want to relock that from the Zoom icon at the bottom of your screen from your little pop-up menu there.
But that's what I see there.
I think we're seeing your screen if you just maybe do the PowerPoint and share it.
Can you all see that?
Yeah.
Yep.
Okay.
Oh, there we go.
Yeah,
It's
that's
still
your
in.
PowerPoint. Yep. There you
That-
go. Perfect.
Okay, very
Perfect.
good. Well, thank you very much, Katherine, and thank you, Keri, for that wonderful, wonderful introduction. So I'm going to just give again, a bird's eye view about Rett syndrome, one of the conditions that Keri has already mentioned that I have been involved in studying for the last 35 years. So Rett syndrome is named after Dr. Andreas Rett, who is a pediatric neurologist from Austria. And this really comes out of his own observations in his clinic. I think the stories that I've heard is that he saw 3 children, girls, in his waiting room with a very unusual hand-wringing movement, and that led to his observation of many other girls. He first reported his findings in 1966. This was in German at a meeting, and then collected 20 to 30 such patients that he again reported at meetings in Austria and Germany. The phenotype, I think his description still is probably the best we have right now. These children are normal early in development, and then there's a stagnation and maybe degeneration. They lose some of their skills, including language, communication, hand use, and they develop a very characteristic stereotyped hand-wringing movement, mouthing the hands or hand washing. This condition was then brought to attention in the Western world by a publication in 1983 by Hagberg and his colleagues. And that was when we all started recognizing this phenotype in our own patients. We knew that it's not that it's a new condition, just that we weren't aware of this. So this image is just a few of my own patients that I've seen over the years. And you see the very characteristic movement, holding the hands together, mouthing the hands. And these generally tend to be girls. This slide gives a kind of an overview of what research has contributed to this field over the past 60 years, the major milestones that have been accomplished. The early part of work was all in trying to define the phenotype, the variation in phenotype among the girls. Soon, case reports were coming in from all over the world. And the focus earlier on was trying to figure out, is this a genetic condition or not? What is the nature of this genetic condition? And defining what the gene is. And once that was accomplished in 1999, the focus became modeling in animals and then developing therapies. Here are some of the key figures in this area. There's Dr. Rett. The next one over is Dr. Hagberg, also from Europe. Dr. Moser, who is no more, he led the effort in characterizing Rett syndrome at the Kennedy Krieger Institute in Baltimore. Dr. Sakubai Naidu is a colleague of mine, and Dr. Moser is also at the Kennedy Krieger Institute. Dr. Huda Zogby, whose work led to the discovery of the MECP2 gene as causative of Rett syndrome, and Dr. Adrian Bird, who were studying the gene way before it became associated with this clinical condition. So the early part of the work was all in clinical characterization and defining diagnostic criteria so that people could decide, whether a patient had Rett syndrome or not. And this was important to support genetic studies as well as clinical trials. So we now recognize typical and atypical forms of this condition, and both of them require a period of regression following apparently early normal development. And then the main criteria are loss of hand use, loss of communication, the stereotypic hand movement, and problems of gait, but other features that are commonly seen, including breathing irregularities. These children tend to hyperventilate and will suddenly hold their breath and might even turn a little blue around their lips. Grinding their teeth, this is what is meant by bruxism. Poor circulation in the feet. Many of these girls will have temperature regulation problems and many other things. So again, as I mentioned earlier on, there was even argument among the experts on whether or not this was a genetic condition or not, whether this was X-linked or not, whether it's an autosomal. So a lot of things were unknown. 95% or more, 98% or more of cases of Rett syndrome that have been described are sporadic. But there were a few familial cases reported, maybe a handful, 7 to 10. And these were key families that led to the discovery of the gene. Now, the genetic studies could not be done as is done in large families using linkage analysis, but these small families allowed the scientists to exclude much of the genome from consideration because they excluded areas of the genome that were discordant between two siblings in a single family. And this is what led to, I'm going to go back once. So this family that I've circled here from Brazil, a family with 3 affected children, was a key to mapping the gene to the long arm of the X chromosome. And after that, it was really a matter of time with the onset, use of with the funding of the Human Genome Project. And at Baylor, they were assigned the X chromosome to sequence. And after sequencing the first 60 genes, they discovered that this was the one that was the cause of MECP2. This was a landmark paper published in 1999 by Dr. Zogby and her colleagues. And since then, there have been hundreds and hundreds of variants in MECP2 that have been associated with this clinical syndrome. There's a database of Rett syndrome mutations that is maintained in Australia with over 300 unique mutations. As I mentioned, 98% are caused by de novo heterozygous variants. That means not familial. And this discovery has led to a great deal of understanding why it is that it affects only girls, and why is it that we don't see an excessive number of miscarriages or male fetal deaths. And it's all because the variant, the mutation, arises on the paternal X chromosome. And, the sperm cells, there is a higher rate of mutagenesis in sperm DNA, and this accounts for many of those things. There are several, recurrent mutations that have, multiple cases reported, and these are listed here. There's also a few males who have been reported with Rett syndrome caused by mutations in MECP2, but because males are hemizygous, their phenotype is much more severe, often causing a severe neonatal encephalopathy. Now, we know a lot about MECP2 now. It is a methyl CpG DNA binding protein. In fact, Dr. Adrian Bird in Edinburgh was studying methyl binding proteins way before the association with Rett syndrome. So this is a cartoon showing the structure of the MECP2 gene. Again, to reiterate what Keri had mentioned about exons and introns, This gene is a very large gene. It has 4 exons, and there are 2 different isoforms of RNA that are generated by alternative splicing. One that includes exon 2, this is called the E2, and one that skips over exon 2, this is E1. They share exon 3 and 4 in common. And this is where 95 or greater % of mutations occur in this region. So these mutations would affect both isoforms of the protein. The E1 is the predominant messenger RNA in the brain. Now, with the discovery of MECP2 as the causal gene, of course, immediately there were several mouse models that were generated and studied, and these replicate features of the human phenotype, including something that might be surprising, which is the hand-wringing clasping movement. This is a hind limb clasping movement that we see in mice. Their mobility is affected and their lifespan is affected. So when you knock out this gene in a mouse, male animals, which are easier to study, usually die by 12 to 14 weeks of age. And then here, just want to highlight a landmark study in 2007 done by Adrian Bird and his colleagues. They devised a scheme by which they could reactivate a silenced MECP2 gene. And they showed that even at a very late stage in the mouse's life, you could rescue the phenotype. These mice, you could rescue them from almost, being, too sick to survive. So this is treated, and this is untreated mice showing the lifespan. So this really was a key study that led us to believe that one could treat this condition and maybe even reverse the phenotype in humans. And since then, the focus has been on therapy development. Now, there are some challenges to both pharmacological, biochemical, as well as gene therapy for Rett syndrome. And I'm just going to highlight a few of these here. One is that MECP2 is a transcriptional regulator. So it modulates thousands of genes. And we first thought that, well, if you just identify all the genes that are modulated, we could come up with a strategy for treatment. But this hasn't panned out. The only 2 things that we understand really well are how it modifies neurotrophic factors like BDNF and IGF-1. The second challenge is that the MECP2 is dosage sensitive. This we call the Goldilocks problem. So overexpression of the transgene in mice causes a neurological syndrome. And there's a human disease called MECP2 duplication syndrome that is also a severe neurodevelopmental disorder. So you can't just inject an AAV virus with a gene therapy cassette into a human being because you have to control how much of the transgene is expressed. The therapy has to be widespread in the brain, and it is required even in adults. So just a transient expression is not enough. It has to last all through the patient's life. Now, there are many, many clinical trials that have tested various drugs, including IGF-1 and cannabidiol, but there's only one of these drugs that has gone on to be FDA approved. This is a drug called trofinetide, which comes out of the understanding that MECP2 modulates the effect of IGF-1, a neurotrophic factor. So this was approved, but is far from a cure. It seems to alleviate some of the symptoms, especially their ability to communicate. Other strategies for treatment, one is searching, these are, again, research strategies, searching for drugs that might promote what's called stop codon read-through. Many of the mutations are nonsense mutations or truncating mutations. So these have been shown to be partially effective in cell and mouse models, but not approved for human use. A second strategy is the idea that one could reactivate the silenced X chromosome. In females, they have two X chromosomes, one of which is inactivated in every cell in the body. And females are actually a mosaic of cells that are expressing either the female Rett patients are a mosaic, where they have some cells that are expressing the wild type allele and some that are expressing the mutant allele. And this is shown in this image here, micrograph image from Dr. Rangasamy, who's my colleague. And this is a mouse brain image where the green cells are expressing the wild type MECP2 protein, and the purely red cells are ones that are expressing the only the mutant protein. So it's possible that you could cure this condition by reactivating the wild type cells in those mutant cells. But the problem is that one cannot do it on a gene by gene basis. And we don't know the effect of reactivating the entire X chromosome. Finally, there are now 2 human clinical trials of a gene therapy vector that has gone through many, many stages of development in animal models. Both of these are in clinical trial. Both of these are an AAV9 vector delivering a MECP2 transgene into the human being, but they have built-in control elements to prevent overexpression, because it's overexpression toxicity that is the major problem. Again, these are both phase 1, phase 2 safety trials that are still ongoing. Now, I'll close with something about what we are doing. Our own research lab is collaborating with Dr. Chatterjee at City of Hope in developing a novel gene editing strategy that does not rely on CRISPR-Cas as a technology. And the problem with CRISPR and Cas is that is the frequency of off-target editing effects that you are unaware of. So this is a new vector. It's called AAV-HSC for stem cell derived. And these have high efficiency of promoting gene editing by homologous recombination. And this is illustrated in this picture by these 2 X's. The idea is you snip and replace the mutant gene with a normal gene. And we have shown that in human patient-derived cells, we can get up to a 20% efficiency of gene editing with these AAV-HSC type of virus, whereas traditionally AAV edits at a frequency of like 0.005%. In this picture, we also show that editing in a male cell can actually restore expression of the protein. So these are both very, very encouraging results. So I think I'll close by summarizing what I've talked about. Rett syndrome is a disorder caused by MECP2 loss of function, primarily in the brain. MECP2 is critical during early development of the brain, during organization, synaptogenesis, and modification of synapses, but it's required in adults. So it's required for us to think and function even as adults. What is encouraging is that restoration of MECP2, even after symptom development, can rescue the phenotype, and you don't have to do it in 100% of the brain cells. So there's a possibility of curing Rett syndrome. We have one compound that is FDA approved for therapy, but it is not a cure. 2 AAV9-based gene therapies are in human clinical trial. These include control elements to prevent overexpression. And there are other strategies that are being investigated, including our work on gene editing without using the CRISPR and Cas technology. So I'm going to finally show, let's see if this works, just the one model that we were working with here in Phoenix. This is a point mutation called the R306C mouse. And so we're going to just put these mice down on the target area, and you'll see the difference between the two. So the one on the right is the normal mouse that is immediately hugs the end of the region and walks around, and the mutant mice tend to freeze, shiver, and don't do too much else. So it's very easy to score phenotype, and this is what we are using now in our gene editing testing at the preclinical level. So I'll close with that and hand it back over to Katherine. Thank you very much.
Yeah, thank you, Dr. Narayanan. I appreciate that. That was a very hopeful ending about what we have to look forward to. So we have some time. We have about 10 minutes, so that's enough time for us to get some questions answered. I think I want to just kick it off with a question I had when I was listening to everyone's presentation is, there's a family out there that is interested in reaching out to learn more about how to participate in what TGen has to offer in your clinics. What's the best way to get in touch with either of you?
Yeah, I think it would be to reach out to me directly
Okay.
at Keri Ramsey at tgen.org.
Okay.
Because we do have a patient registry, but then this way you get direct access, and I usually respond within a day or two.
Okay. And are there any criteria that people need to know about, or do you just review cases one at a time as they come in?
Yeah, I mean, we have plenty of patients that are just self-referral, so it doesn't always have to be through their physician. And a lot of times the process is working with the families to obtain outside medical records and get a pretty in-depth, health history, family history from them to determine if they're eligible for our studies.
Okay, so that sounds like a good first step, just to reach out to you. Okay.
Mm-hmm.
And this question can be for both of you. So the question is from Dr. Sklar. He said these therapies are very exciting, but he has two quick questions. The first is with sickle cell treatment. How do we shut off the sickle hemoglobin so it does not compete with the fetal hemoglobin?
Yeah, I think from what I've read, the goal is not to target the sickle cell gene, but to dilute the hemoglobin with that fetal hemoglobin, and that itself prevents the sickling. So I don't know if, I think at this point that's providing enough benefit. I don't know if they're also trying to do that as well.
Yeah, okay. That makes sense though. And then the second question is on everyone's mind, and that is the cost. I know that I've seen some articles and some publications about that, and I know it's been in the media not too long ago. And so because it's such a high cost, how do you think it's going to affect, I guess, thinking about all the healthcare costs that we're dealing with in this country? Do you think it's going to be provided to all patients with sickle cell disease, and how would that impact the Medicaid budget if we consider the fact that many people don't have traditional health insurance? And I don't even know, maybe you could comment, I don't even know if health insurance covers it. So maybe that's something you might have some knowledge about.
Yeah.
Maybe I can try and answer that question, at least for SMA. I'm not sure I can answer that for sickle cell disease. Mm-hmm. But this was a big concern because I think initially the list price for the AAV9 gene therapy for SMA was well over $2.5 million for a single injection. But I think the way the price, again, how pricing is determined by the manufacturer is still a mystery to many of us, but they probably factor in the costs that are borne by the patient over their lifetime. And so I think I've heard that the insurance companies have come up with various ways of reimbursement, and it might not be all at once. They may not get a single check, but it might be amortized over the life of that patient. So many years, they might get, the manufacturer might get a discounted fee, but it might be distributed over a long time, not just as a single payment. And I think insurance companies are covering the cost of the SMA gene therapy, definitely here in Arizona.
That's good news. Yeah, and it's like any other rare disease when you start getting treatment. Like if you think about hemophilia back in the day and the cost of the medication over time, it gets figured out, but it takes time, and it takes the insurance companies getting on board with how that's going to, as you just said, be allotted out. So I think it's still kind of new to figure all this out. But I know there always is a lot of questions from people about how it's going to get covered and what's going to happen in that regard. So we'll keep an eye on that. And it is impossible to figure out what insurance companies are going to do. I wholeheartedly agree with you on that one. And the next question is, do you expect AI to speed up genetic test processing? Or is it already being utilized is it in process to reduce the cost, or where are we at with that? Is it not there yet?
Yeah, I mean, AI is definitely being used for variant interpretation. So, after a person has genome sequencing, you get back, thousands of changes in the DNA, and AI is being used to identify if those changes are significant and going out into the public literature and pulling those papers. And it can, identify like if you have a specific variant, has it been seen before in certain individuals in the publications, and it'll pull that information out. So it's cutting down, reducing the amount of time that humans had to do literature review. So that's just like an example of one use of it. And then it's also being used for drug development as well. So we were working with a group that was using RNA sequencing data, and they had an algorithm where they were testing that RNA data against multiple drug compounds, and they had actually come up with a potential therapeutic for Rett syndrome, and it's now being put in clinical phase at Vanderbilt University. So this is all from AI software.
That's good use of AI. I approve of that. I have mixed feelings.
As long as it doesn't replace us, right?
Yeah, right. I know, I have mixed feelings about that. Keri, I think this one's for you. What do you think are some of the biggest challenges families face when seeking a diagnosis for a rare disease? And then also, what are some of the best resources available to support these families?
Oh gosh, there's so many challenges. Fortunately, like the barriers to testing have come down the last few years. It used to be that not everyone had access to genetic sequencing. And I think what's really hard is that even with this type of technology, we're still not able to diagnose 50 to 60% of these families. And we'll have families where there's multiple children that are affected, and you're like, this has to be genetic, or genetic and environmental. And so I think that's still a huge challenge, is these undiagnosed families. And then even when you receive a diagnosis, it doesn't always lead to treatment. Sometimes you're still treating the patient based on their symptoms. And so I think, and what happens is these families, they'll get a diagnosis, and then all of a sudden now they'll be raising funds specifically for that diagnosis, as opposed to broader treatments for multiple disorders. And so it kind of can spread the research and the funding, it can make it thinner across multiple disorders. And sometimes there's disorders where families, they're really driving the research, they're driving the funding. And others just don't, the families don't have those types of, that type of time to put into it. And with families, I would say, keep in touch with your clinicians, your geneticists, because there's always new technologies coming out, and what's being covered by insurance is constantly changing. So even though you may have been denied like 5 years ago specific testing, go and see your doctor. Make them aware that you're still looking for answers, you're still looking for therapies. I think Global Genes is a great website. There's NORD. There's some websites out there for rare disease. I don't know if Dr. Narayanan, you have anything else you want to add to that?
I think, families are driving this, and, I think we can help. If people reach out to us, we can help them. There's the undiagnosed network, but I think sometimes it's very hard for families to get through to these places. They need some guidance. But it's a tough battle.
Well, I appreciate both your responses, because essentially you're trying to empower the families, are empowering themselves to reach out. And if they have you as their advocate and their point of contact, then it gets easier. So I really appreciate that response. And I hope whoever's listening and needs that information can do that. Follow up with your doctor, reach out to Keri, and that would be great. We have time for one more quick question. It's a very interesting question, so I want to get to that one. This is probably for both of you. I'd like to hear both your perspectives. So first of all, great presentations, this person indicated. Are there any plans to include testing for these rare variants in prenatal genetic screenings?
Okay.
You take this one.
All right. Okay, wonderful. Well, I think a few years ago, I was thinking the same thing, that, of course, Rett syndrome is an example of a de novo mutation causing disease. So we would not be able to assign a risk to a couple before they ever had that child. But SMA is a good example of an inherited, neurological condition that was previously fatal. So we, again, the parents would have no knowledge that they are carriers for these common mutations until they have an affected child. So I was thinking that we have all of this technology to make diagnoses in children who are affected with some neurological syndrome. Can we apply the same genome sequencing methods to do preconception genetic screening, at least to identify, risk alleles in the parents and counsel them about that risk. So we did a pilot project that was funded by the Flynn Foundation. And, this is published. It's a very interesting finding. And we've tried, I think we are trying to see if we could, population-based screening for something like SMA in the state of Arizona might make a difference, especially when the costs are so high. Imagine if we could identify and counsel, but the problem is it's not enough just to do the testing. You have to counsel the families beforehand
And
and after the test results are obtained, so that they understand, they know what are their options for reproductive decision making. So they complicated, but I think if we invest these resources, we will make a difference in the world, and maybe reduce ultimately the cost in dollars as well as emotional cost to these families.
Yeah, well, thank you for that. I'm sorry I had to cut you off. We're over time, so I want to thank our presenters for both of them for some outstanding information and lots to think about, lots to consider in the future. So thank you all for joining us. We are, as I said, out of time for today. If you've not already done so and you're still with us, please register your attendance using the self-check-in link posted in the chat so we have an accurate record of who attended today's webinar. We encourage you to follow the College of Health Solutions on social media so that you can learn about other opportunities like this one, and you have all the links up here on the screen right now. And then our next series will continue on March 20th, when we will be discussing the impact of the Rett microbiome on overall health. Details and registration will be available at asuhealthtalks.com. And thank you again. Thank you to our presenters for a really outstanding hour of information. Take care, everyone, and have a great rest of your day.